And this will be the last class on development of brain, spinal cord, and pituitary gland. So here we'll deal with the development of prosencephalon, which is divided into diencephalon and telencephalon. The prosencephalon is divided into cranial telencephalon, which gives rise to cerebral hemisphere, and caudal diencephalon, which forms the thalamus, hypothalamus, optic vesicle, and posterior pituitary pineal gland. So diencephalon has a cavity called third ventricle. So it has roof plate and alar plates. Roof plate is made up of ependymal cell layer, and it is covered with vascular pia. Portion of ependymal layer with tuft of capillaries from pia mater invaginates third ventricle, forming choroid plexus of third ventricle. The caudal part of roof plate forms pineal body. You can see here this is the roof plate. You can see the caudal part of the roof plate forming the pineal body. In the floor of the diencephalon, you can see there is formation of the mammillary body. You can see the infundibular process which forms the neurohypophysis, and you can see the optic chiasma. So development of epithalamus. The roof plate of the diencephalon gives rise to epithalamus, which gives rise to pineal gland, habenlar liquid, and habenlar commissure, which is concerned with the olfactory pathway, and a small posterior commissure connecting epithalami also develops caudal to the pineal gland. Alar plates. Alar plates form lateral walls and floor of diencephalon. The alar plates will form three zones separated by two sulci, hypothalamic sulcus and epithalamic sulcus. The three zones are hypothalamus, the lower part, thalamus, the middle part, and epithalamus, the upper part. Thalamus gradually bulges into the cavity of third ventricle and differentiates into thalamic nuclei. Hypothalamus gets pushed caudoventrally, that is anterior-inferiorly, due to enlarging thalamus. You can see here, this is the thalamus and this is the hypothalamus separated by hypothalamic sulcus. You can see the corpus striatum, which is being formed in the telencephalon. You can see the choroidal plexus coming here, and you can see the telencephalon forming the cerebral cortex. At 8 weeks, you can see the enlargement of the choroidal plexus, and you can see the enlargement of lateral ventricle, and you can see the corpus striatum. You can see the thalamus and hypothalamus separated by the hypothalamic sulcus. You can see clearly the well-developed thalamus and hypothalamus separated by hypothalamic sulcus, and you can see the inferior horn of lateral ventricle. You can see the body of the lateral ventricle. You can see the fornix, and you can see the choroidal plexus. You can see the corpus callosum, which is forming the commissural fibers. You can see the claustrum and lentiform nucleus and the caudate nucleus and the tail of the caudate nucleus. You can see the hippocampus here, the inferior horn. So in the floor of the cavity of the diencephalon, a group of neurons assembled to form the mammillary bodies, I have shown in the earlier picture. Then on the 22nd day, in the region of hypothalamus, the lateral wall of diencephalon forms optic sulcus, which later becomes optic vesicle, forms the optic cup, so it forms the retina and muzzle of iris. So from the floor of diencephalon, a downward growth develops that forms the infundibular process. Later, the infundibular process develops into posterior pituitary. So now we'll see the development of pituitary gland. It's an unpaired endocrine gland, also called as hypophysis cerebri, located in bony fossa called sella turcica of the sphenoid bone. Two major parts, adenohypophysis are antipituitary and neurohypophysis are postpituitary. Antipituitary develops from Rathke's pouch that arises from ectoderm of stomodium. Anterior lobe consists of pars distalis, intermedia, and pars tuberalis. The cavity of the Rathke's pouch forms the intergranular cleft. Neurohypophysis develops from the infundibulum, from the diencephalon, from the floor of diencephalon. You can see here, this is the diencephalon. You can see the ectodermal thickening, the Rathke's pouch. You can see the developing Rathke's pouch, and you can see the infundibular process. You can see here the infundibular process. This is the Rathke's pouch from the stomodial ectoderm of the stomodium. You can see the infundibular process coming from the floor of the diencephalon. So this forms the posterior pituitary, this forms the anterior pituitary, and this persists as the cleft. You can see here this is the intragranular cleft. You can see the anterior wall, posterior wall, and the cleft. And you can see the regression of the Rathke's pouch. This is the infundibulum. So this is the posterior lobe formed from the infundibulum, and you can see this is the anterior lobe formed from the anterior lobe and intermediate lobe formed from the Rathke's pouch. You can see the site of regression of pharyngohypoglossal stalk. If this persists, it results in craniopharyngiomas. You can see the pars intermedia, pars distalis, and pars tuberalis, which develop from the Rathke's pouch, and you can see the infundibular stem and posterior lobe developed from the infundibulum. So Rathke's pouch, anterior wall, pars distalis, pars tuberalis, the cavity, intergranular cleft, posterior wall, pars intermedia, anterior pituitary. Infundibular process, infundibular stalk, and pars nervosa, posterior pituitary. Blood aspects. craniopharyngiomas. So remnants of the Rathke's pouch gives rise to craniopharyngiomas, accessory pituitary gland along the course of the Rathke's pouch. And you can see the pituitary agencies are hypoplasia. Then pineal gland, also called epiphysis cerebri, it's also called as third eye of human body or principal seat of soul, develops as an evagination from diencephalon in the roof of the third ventricle, which I have already shown in the diagram, made up of pinealocytes, modified neuroglial cells. Pinealocytes secrete melatonin hormone. Melatonin inhibits the secretion of gonadotropin-releasing hormone. Development of cerebrum. Prosencephalon consists of two lateral outpocketings as cerebral hemispheres and a median lamina terminalis. So cavity of the prosencephalon forms little ventricle. Especially this prosencephalon is, I am speaking specifically about the telencephalon. Telencephalon consists of two lateral outpocketings as cerebral hemispheres and the median lamina terminalis, which represents the anterior neuropore. So the cavity of the telencephalon is the lateral ventricle, right and left. So development of telencephalon during first 2 months. It has superior pallium and a basal part. You can see here. So this will be the superior pallium and the basal part will be in the center. You can see how smooth it is. So gradually you can see in the 6th month you can see some part of sulci and some gyri. So you can see prominently the lateral cerebral sulcus. So here in the 9th month you can see completely developed telencephalon with the central sulcus, lateral sulcus, and all majority of the sulci are formed. So initially the surface of the telencephalon is like this, but at the end the surface looks like this. So gyri and sulci are formed to accommodate the rapid growth of the telencephalon or the cerebral hemispheres. So cells from the basal part migrate into pallium that forms cerebral cortex. Remaining cells of the basal part form the corpus striatum. So this corpus striatum will have this caudate nucleus, lentiform nucleus, claustrum, amygdala. The junctional zone of two pallium, the cerebral cortex, is very thin and gets invaginated by choroid plexus forming the choroid fissure. The pallium grows and gets differentiated into allocortex and neopallium. Allocortex is the old cortex, neopallium is the neocortex. 90% of the cerebrum is having the neopallium. Allocortex is having archaeopallium, oldest cortex, and middle evolved cortex, the paleopallium. So efferent and afferent fibers form the cerebral cortex, form the internal capsule. So you can see in the development of cerebrum, after the 2nd month, the neopallium overgrows and compresses the allocortex. In the floor of the cerebral hemisphere, group of neurons condense to form the striated nuclei. These are the corpus striatum. These striated nuclei get traversed by fibers of internal and external capsule. The striated nuclei differentiate into the following: the caudate nucleus, putamen, claustrum, globus pallidus, and the amygdalic nucleus. Then the cerebral cortex grows in various directions to form various lobes: the frontal lobe, occipital lobe, parietal lobe, and temporal lobe. You can see here. You can see here the telencephalon developing into the cerebral hemisphere, and this is a diencephalon. And here you can observe certain commissures are forming, that is connecting the two cerebral hemispheres. You can see here. This is the anterior commissure and this is the corpus callosum, which is not completely formed, it is just appearing. And you can see the havenular commissure and you can see the posterior commissure. Now if you see here, you can see the completely developed corpus callosum, and you can see the anterior commissure, the posterior commissure, havenular commissure, and there is also an additional commissure called fornicial commissure connecting the hippocampi of both sides. You can see the anterior commissure connecting two temporal lobes, then corpus callosum connecting right and left cerebral hemispheres, hippocampal commissure or the fornicial commissure connecting right and left hippocampus, posterior commissure. It is essential for light reflex. Then havenular commissure connecting the havenular nuclei, then optic chiasma. You can see here anterior encephalocele, there is a defect in the anterior part of the skull. Through that the brain is protruding out. And you can see the anencephaly, the failure of closure of the cranial neuropore resulting in defective development of the part of the brain and skull. Holoprosencephaly, the midline structures are not formed well, in which the loss of midline structure results. Thank you.